cells, and we can see the mucus cells, but not the different types of glands. So what gland would this one? This one already is just obviously simple for you, sublingual. So this one, which is, you can tell, is serous. The purple is going to be carotid. And this one, which is a mixture, is similar to the one, identical to the one I showed with lecture, which is submandibular. So the microscope slides, or some of the microscope slides have some that have all the samples on them. So remember I talked about the serous wounds, where you have the crescent serous secreting on the mucus. And so you can see that in the submandibular and the circular zone. All right, so we're starting with, and I found this picture. We're going to tell you guys about the surgery that they did in the C-section and took the uterus out. And this is 21 weeks.
Wow.
And just as he was closing up the uterus, the fetus reached out and grabbed the finger. So that you can notice ovary down here. All right. We went through stomach. Oh, here's the, I hadn't shown these yet either. Remember we talked about the junctions? Between, there's an abrupt transition from the esophageal epithelium of its mucosa to the gastric. And so though it's low power, you can see a distinct difference between the stratified squamous and this is simple columnar. You probably can't see individual columnar cells. Point of awareness, these are the gastric pits and the gastric glands. When you're looking at the microscope slides, because next week we'll have your histology quiz, when you're looking at the microscope slides, remember the gastric pits, when they're really deep, as they are in the pylorus of the stomach, can be confusing with the villi of the small intestine that we're going to talk about today. In the fundus, they're more shallow, even more shallow than this. This is the cardia. And so be aware that you're not looking at villi here, you're looking at gastric pits. So when you're looking at your microscope studies, you want to make comparisons, get slides and make comparisons between those components. Okay, with the comparison we made, you have to take the difference between stomach and esophagus versus anal canal and rectum. So we're going to get through rectum today, and these are goblet cells. Remember, about approximately 25%, but they look like 100 when we look at this histology view. Okay, so just these might look like the neck mucus cells of the stomach, but those are found only in the gastric pit mucus cells, whereas the goblet cells in the colon and even in the intestine are going to extend all the way down to the gland. All right, so we're looking at the small intestine today, and we had started off with a series of sphincters. I'm going to kind of put those over here on the side. So what was the first sphincter that food passes through when we were talking about our digestive tract?
The one in the esophagus. Yes.
Nope.
Your ventricularis oris.
Notice my face? Amy caught it. So the mouth, essentially, orbicularis oris, which is skeletal muscle, and we end with skeletal muscle as well, the anal canal. And then between the stomach and esophagus, or esophagus and stomach, we have the cardiac sphincter, or gastroesophageal. I just feel like writing that all over again. And what was the last tubular region of the stomach? Remember what that part of the stomach was called?
The one?
No, stomach.
Pylorus.
The pylorus. So that next sphincter then between the stomach and small intestine is the pyloric sphincter. All right, so we have two more sets of sphincters to look at between small intestine and colon and anal, the anal canal. So our next structure, we're going to look at its components first, and then the G-pad, or the basic plan of tissue outline. And this broken down into three regions, right? The duodenum, the jejunum, and the ileum. They all characteristically have villi, and remember, this is our basic tissue organization. What our plan is based on is small intestine, and then our submucosa, mucosa, muscularis externa component, and then we made additions or modifications to that if we were in the mouth or the esophagus or the colon. So I'm going to draw a small portion of the stomach here. And anatomically, there is not as much room as I'm going to create between the duodenum and the stomach, which is to give me more room. The first portion of the small intestine is known as the duodenum, or you may pronounce it duodenum, as long as you spell it correctly. The duodenum is retroperitoneal. Remember we talked about primary and secondary retroperitoneal organs. So if it was intraperitoneal and then fuses to the posterior abdominal wall and loses the peritoneum of the parietal and visceral components and becomes retroperitoneum, that's a secondary. And that's what happens to the duodenum. It rotates over and is fused to the posterior abdominal wall. Then right at this point here, see this knob? That knob is where it becomes intraperitoneal and is the indication of the beginning of the jejunum. This is where we see the beginning of our root of the mesentery. And that will be where I would tag it in lab on a quiz or an exam, because once we're past that point, as you can see on the image, it becomes rather like a coil of snakes and it's a little difficult to tell exactly where we are. The most distal portion then is going to attach to the cecum via the ileocecal valve, and so this portion is the ileum. Okay. These two segments are intraperitoneal. Anybody remember? I've mentioned them just in the last 3 minutes. What are the two mesenteric structures that we associate with the small intestine? Fatty apron is stomach and large intestine. That's what it attaches to. The lesser omentum and the greater omentum are associated with stomach, liver, and colon.
The
Mesentery proper, and then the mesentery proper, remember I just indicated, is attached to the posterior abdominal wall via the
Root of
the mesentery. We don't have any lab models that show either of these very well, so likely to be diagrams, all right, or a sagittal section that I drew on the wall for you is going to indicate this. Now, we're going to talk about their function on Thursday, but I want to just put up here on the board briefly a summary of their function. Notice how the duodenum is a short segment. That's not going to be very much surface area for digestion, and certainly if we don't have digestion occurring, we don't have absorption occurring. So what can you imagine as the purpose of the duodenum? Pardon? Exactly. It's a receptacle. And it receives items from 3 organs. You can already see that it's receiving from the stomach. Okay? So it's going to receive chyme from the stomach. That's what we call the mechanically digested food from the stomach. As we'll see later today, it's going to receive bile salts from the liver. And we'll be looking at the purpose of bile salts in lab on Thursday. Anybody know what the next organ is that I haven't drawn on the board?
Gallbladder?
Well, that stores the bile salts.
The pancreas?
Pancreas, all right? So it's going to receive digestive enzymes and bicarbonate ions from the pancreas. And so it becomes an area where that material then is mixed, and then finally it moves past through the duodenum into the jejunum. Jejunum, if we were to pick a particular function that's primarily for the jejunum, is chemical digestion, although this will continue into the ileum. And then the ileum is for absorption. Not that both don't occur in both places, but as you'll see when we do the physiology of digestion, the pancreatic enzymes need to do the intermediate stage, and then finally the intestines do the final stage. Remember when we looked at the blood supply review? The small intestine is supplied arterial blood and nutrients by the branches of the superior mesenteric artery. So if I were to use this as a lab quiz or a lab exam image, this mesentery structure here is the mesentery proper. Okay, it's that kind of skirt that we can spread out that would contain the blood vessels, and this location along here is where we have the root of the mesentery. Okay, now remember what I told you about the difference in blood supply as far as jejunum and ileum are concerned? So we can see some changes in the villi, but without doing microscopic structure. When you look at these arcades, the single arcades are jejunum, and the multiple arcades are ileum, or that increased ability for absorption, probably. All right, we're going to look at the mucosal pattern. And the two distinct, we've already have gone over our basic standard, all right? So, may I raise the board? You guys have that diagram that's on the board?
Hold on.
Okay. You're set? Guys, remember what GCAT stands for? General Plan of the Alimentary Tract. So again, this is our standard, so we're not going to make any changes, but I want to illustrate those components for you. Remember when we went over the mucosal plan for the stomach? We had almost looked like castle walls with crenellations where we had gastric pits and gastric glands. That was the stomach. So in the small intestine, we add surface area by adding villi and intestinal glands to replace the gastric glands. So when we look at the mucosa, we started simple columnar epithelium with the stomach. Remember, we only have 2 types of epithelia in our digestive tract. So we continue that with simple columnar epithelium in the small intestine. Microvilli or cilia? Microvilli. Now we didn't see goblet cells in the stomach, but we do in the small intestine. So goblet cells are present. Certainly our lamina propria, our loose irregular connective tissue. And finally we end with a muscularis mucosa. So let's take a look at those intestinal villi. Simple columnar epithelium in the intestinal glands. With scattered goblet cells. Microvilli. So this entire structure is identified as a villus. Now we had specific cells in the stomach that we call the chief cells, and what were the cells that make hydrochloric acid? Use hydrochloric acid? Huh? Striatal cells. Okay, and cells that release the hormone gastrin. G cells or enteroendocrine. And so we don't have that great variety. Almost all the cells that the cells that aren't goblet cells are going to be releasing digestive enzymes. By releasing, we'll come back and talk about that on Thursday. They don't secrete them into the lumen of the small intestine, but those digestive enzymes remain bound to the microvilli. All right, so maybe rather than secreting, synthesizing would be a better term, because that chemical interaction takes place on the surface of the microvilli. What structure, lymphatic structure, you guys remember me pointing it out to you? In the center of the villus is a lymphatic capillary. Remember, those are blind endings. And we have a specific name for lymphatic capillaries that are found in the intestine. Lacteal. So you need lacteal for full credit. Lymphatic capillary would get you partial credit. Okay. In addition to the lymphatic capillary, we have our cardiovascular system, our blood capillaries. Because this is loose, irregular connective tissue. So we're going to be providing a capillary system. So, again, we'll review this when we get to the physiology of it. But in our blood capillary system, what are we picking up?
Amino acids, glucose.
Okay, so simple sugars and single amino acids. And those are going to go into our venous system, the superior mesenteric vein. And what vein does that drain into?
Portal vein.
Portal vein. So all of these nutrients are getting into the liver via the portal vein. What is going to be picked up by the lymphatic capillary, the lacteal?
Lipids.
What are we missing from our organic list of nutrients here. The proteins,
Lipids.
lipids, right? So lipids, because of their non-polar covalent format, are not very soluble in the water of the plasma, which is polar, and they are then covered with a coat of proteins that allows them to form interactions with the charges on water molecules, and that makes them too large to get into the blood capillaries. But remember, whole cells can get into the lacteals, or white blood cells can get into the lacteals because of the gap between the squamous cells that form that. And so our lipids, fatty acids, triglycerides, and so on, are going to be picked up by the lymphatic capillary, and that's why that is of significance in our digestive system. Is that going to go directly to the portal vein?
No, it comes out in the.
We go to the cisterna chyli and the thoracic duct and the subclavian vein to the heart, and then it gets pumped out with the blood. So the hepatic artery is going to be taking this material to the liver. So portal vein and hepatic artery are going to be rich in those particular nutrients. All right, so just like the rest of our mucosa, we have a lamina propria, our loose irregular connective tissue, and we end up mucosa with a muscularis mucosa. We'll come back and talk about the villus in just a moment when we talk about how we increase the surface area for the small intestine for enzymes and absorption. But as we look at the mucosa here, we'll move on. The next layer is submucosa. So I'm going to erase this and write submucosa if you want images. So we'll move our muscularis mucosa up here a little bit higher. So our standard tissue for the submucosa is just a dense, irregular connective tissue. But we have specializations. So this previous couple of slides here show a specialization in the first 3 or 4 inches of the duodenum. And that C-shaped curvature I showed you, where we had the entrance. So our bicarbonate ions from the pancreas really didn't get in until about here, but we've got all this acidic chyme coming from the stomach. So to protect the cells of the duodenum from all that acidity, because that's not going to get neutralized until we get the bicarbonate ions from the pancreas, we have these submucosal glands. Again, they're not goblet cells, they're mucus secreting cells similar to our sublingual gland. And so these are specialized high concentrations via a duct, similar to the distal esophagus, secreting additional mucus that protects the surface of the small intestine, well, specifically the proximal duodenum. So on this image here, these are intestinal villi, and these, you can see how dense these glands are. They look like they take up the entire submucosal region. So submucosal glands in the proximal duodenum. And what do we find in the distal ileum? Tire patches. Okay. So these would be lymphatic follicles without a capsule. They're not lymph nodes, they're lymphatic nodules or lymphatic follicles as I prefer to call them. Up here, we would have our mucosa with the intestinal glands and the villi, and we would have diffuse lymphatic tissue throughout the entire gut region. But we have an increase of lymphatic follicles in the ileum. Anybody remember why?
Because it's so close to the rectum,
We'll
back
call
very
it.
good.
Right, so we have the normal flora of the large intestine, and the ileum is attached to the large intestine, open to it via the ileocecal valve, and so we have then that additional component of increased lymphatic tissue. So the ileum is going to show the Peyer's patches as a modification of the submucosa. Muscularis externa is next. Well, before we get to that, let's go back and look at some images of the villi. Got some here, but so this is a scanning electron micrograph of villi. Again, these are above the surface area. If we consider the surface area of the floor, we would still have gopher holes, which would be our intestinal glands, and then we would have mounds or finger-like structures rising up above that. These are not moving, all right? When I say finger-like, they're not going to be doing this in motility, but function to increase surface area. So here would be a single intestinal villus, and the old name for intestinal glands was crypt of Lieberkühn. Much more beautiful a name than intestinal glands. But those are going to be the source of the cells in the intestinal glands. I should have covered this before I did the submucosa. Down at the base of the intestinal glands, we have enteroendocrine cells releasing hormones from the small intestine. Why would these endocrine gland cells, which are not the plant cells by the way, but why would these enteroendocrine cells be at the base of the glands rather than out on the intestinal villi? They're at the base of our gastric glands as well.
Back down to the acidity?
Pardon?
Is it so they're not exposed to the
No.
acidity and
No.
Is it so the whole cell gets the
What does an endocrine cell do?
synthesized hormone?
Synthesizes hormones. Does that need to get into a duct or out into the lumen of the small intestine? How are hormones distributed? In the blood. So being at the base of the intestinal glands is going to have greater access to not just the capillaries, but immediately into the larger blood vessels as well that the capillaries are associated with. So it doesn't need to go into the lumen. It's endocrine, not exocrine. In addition to that, I don't know if you can see these cells. The Paneth cells look a little bit like mast cells with these granules. It's not histamine, but these are defense cells. These are antibiotic-type materials that are released to protect the small intestinal cells, because they're doing absorption, are a little bit leakier, a lot leakier than our stratified squamous cells. So as a protection, in addition to the diffuse lymphatic tissue, we have these Paneth cells that secrete antimicrobial proteins. Okay, sorry, I should have done this when we did the mucosal structures. All right, yes.
This is the mucosa of what? Of the small?
Well, this is the submucosa down here. So this would be the intestinal gland. Let me pick up the histology model here rather than diagramming that structure. So this is our mucosa here. These would be the villi, and these would be the intestinal glands. So the Paneth cells are down at the base of these cells, just above the muscularis mucosa, that you can see here. And then the submucosa starts down here. So submucosa would be down here, and this represents the submucosal glands that are in the that are in the model.
So that was what we were drawing, because when we drew the enteroendocrine cells?
Enteroendocrine cells are not PANA cells. The enteroendocrine cells of the stomach release the gastrin hormone. In the small intestine, there's several different hormones
Okay.
that are released. Okay? Again, before we move on to the muscularis mucosa, notice, sometimes students skip over this, if I were to draw a cross-section